working. We'll still take breaks every hour, so we will give ourselves some screen-free time. And, you know, between kind of each chunk, we want to have, you know, time for your questions and things like that. So it'll feel just like a normal session. All right, so I'm going to share my screen. I won't be able to see your hands or questions necessarily. So if I am missing one of you, please let me know. All right. Y'all can hear me still, correct?
Yes.
Thank you. Okay, awesome. All right, so you can see my screen. I'm going to just go through some of the resources we have for today, and then we'll get started. So on our page here, you can kind of see the topics that we're going to refer to or go through. We're going to focus first on aerobic respiration, carb metabolism. We're going to kind of go through all of those main metabolic pathways to digest glucose and its counterparts. The second hour, we will go into cholesterol synthesis, lipid metabolism, and then probably start moving into some urea cycle conversations. And then the last hour, it's kind of a popcorn hour. That's initially what I had it called. But this will be for some key vitamin pathways. We're going to talk about heme synthesis and really anything else that we haven't gotten to thus far. These page numbers are associated with this large PDF that I posted. It's like a 105-page PDF that we'll be referring back and forth to. But besides that PDF, there's also some other resources I want you to be aware about. So in this biochemistry resources folder, there are a ton of things I've posted. These are like traditional biochemistry PowerPoints. I would only review these PowerPoints if you're struggling on grasping some of the concepts that we're going through relatively quickly, because we're going to focus on high yield. I'm going to walk through some of these pathways, but we're not going to talk about all the ins and outs of the biochemistry of it all. So if you're struggling, these are PowerPoints that will have way more in-depth than you'll need from more of a biochemistry course perspective. But they're in verbiage that's a little bit sometimes easier to understand. I'm going to mainly be referring to these kind of 3 PDFs, as well as this just general biochemistry PowerPoint. And this general biochemistry PowerPoint will kind of get us started. It has kind of the major pathways we're going to review on each slide. And then the PDF that I'm referring to is here. You'll see that the page numbers are going to reference the pages of the PDF. Now, how can you not pass out and fall asleep during this online virtual biochemistry session? My suggestion is to follow along and actually fill out this table. And this is really, at this point, for high yield study, I encourage you to focus the majority of your biochemistry that's not physiology based. And what I mean by that is there's some biochemistry that's the enzymes involved in key physiologic pathways, right? We've been kind of talking about some of those as we go through, specifically our adrenal hormones or adrenal steroid hormones that we've discussed. But when it comes to the actual biochemistry, the metabolism of the body, this goes through where the tissue, these pathways are located, the cellular compartment, if appropriate, substrate that it starts with, the major key enzymatic step, often referred to as the key regulatory enzyme, any products that are associated, any specific regulators, intermediates, and what's the energy either generation or requirement. Dr. Megan Taylor initially made this essentially table back in the day, and we have been using it since because it is really such a great overview of kind of how you can organize this information. You also can see here that this is a started one for you. So if you want the fully blank versus one that kind of gives you an idea of what this could look like, as well as the answer key with everything filled in. And of course, you could make this a lot more expansive, but we really try to focus in on the most important components of these pathways. So these are the things I'm going to be referencing. I really think that it's helpful to follow along with this component. I will also give you some like mnemonics and important acronyms and things as we go along to try to help with your study. And if anything is helpful that I say, I can always type it into the chat too. So we're going to start right at the top with our glycolysis. So I'm going to go to our biochemistry PowerPoint, the ins and outs of what you need to know. So glycolysis is where we're going to get things started. Why? Because this is the major pathway for us to get fuel into the body. This is how we digest glucose. that monosaccharide and actually convert it into eventually energy in the form of ATP. Glycolysis is a pathway is going to occur in all tissues. It's going to occur within the cell in the cytosol area of the cell. And we start with glucose. Now we can shunt in galactose and fructose into this pathway. And I'll talk about where those might come into play a little bit later. But we're going to primarily focus on glucose for now, as that's our main sugar that we're going to be utilizing to get energy. The key enzymatic step, or the key regulatory or rate determining enzyme, is PFK1, or phosphofructokinase 1. And so we can see here, let me find our PFK1 if I can see it. There we go, our phosphofructokinase. So if we look at this picture, and we'll look at a couple different examples, phosphofructokinase, PFK, is really taking over this third step from fructose 6-phosphate to fructose 1,6-bisphosphate. What is this enzyme doing? Essentially, it's adding a secondary phosphate group onto our first carbon. So we can see fructose 6-phosphate right here. Our 6th carbon already has a phosphate group on it, that P, and then phosphofructokinase takes this one carbon labeled 1, and it's going to add a phosphate group to it. And this key regulatory enzyme step is important. It can be upregulated and downregulated from different things. AMP, which is our breakdown from ATP, right? Adenosine monophosphate, will upregulate this process. Fructose 2,6-bisphosphate, which we'll look at as a molecule, will upregulate this process. If we have a lot of ATP in the body, it's going to downregulate this process because we don't need more. Or if we have too much citrate in this process, which we'll see that connection when we get into our Krebs or TCA cycle, that will downregulate this process. So those are kind of some key points in regards to rate limiting enzymes for glycolysis. Looking at another enzyme that could be important to remember, if you're going to remember one other enzymatic step, it'd be hexokinase, which is your enzyme that is essentially the first step in this process. You could say that this is the step that commits glucose to going down this pathway. I have hexokinase and glucokinase here because they have very similar actions. In fact, they both add that phosphate group onto our 6th carbon, which we can see here. So they're both going to take glucose, and they're going to, we have the 6th carbon right here, they're going to add a phosphate group to it. So we can see that phosphate being added. Hexokinase and glucokinase both do this. The major difference between the two is glucokinase is primarily in the liver. Hexokinase is in the majority of other tissues. So if you're going to put something to memory, remember that glucokinase is going to do this first step in the liver. Hexokinase is going to do this first step everywhere else in the body, because all tissues can undergo or can perform glycolysis in the body. Okay. What are some other things that can help regulate this pathway? You can see that insulin is going to upregulate this pathway, as well as, like I already mentioned, the fructose 2,6-bisphosphate and AMP. Why insulin? Well, if we have, let's say we have lots of sugar in our blood and our body, we're a hyperglycemic state and our body is needing energy or wants to get some of that sugar out of the blood, insulin is going to be secreted from the pancreas. That's going to upregulate our GLUT transmitter or transporters, take more glucose from the blood into cells, and then this process will increase. If we have more glucose in the cells, more glycolysis will take place. So insulin, by pulling glucose out of the blood, upregulates the process of glycolysis. You could say that glucagon does the opposite, but glucagon is really going to govern a different process that we'll talk about here in a moment of harvesting glucose later. Let's see here. When it comes to energy production, that's one thing for this whole pathway we'll be discussing. Energy production, we get 2 pyruvate at the end. We get 2 ATP, which we'll see those steps here in a moment, and 2 NADH. And so as we track kind of aerobic respiration, it's important that we understand what are the energy requiring reactions versus the energy creating reactions. And so we can look here and see that there are several different kind of energy requirements all highlighted in yellow. Our two energy requirement reactions, meaning that we have to put ATP or energy into the system to move it forward, are our two rate limiting enzymes. So hexokinase and phosphofructokinase, or glucose to glucose 6-phosphate and fructose 6-phosphate to fructose 1,6-bisphosphate. So these two reactions are going to cost us 2 ATP per glucose molecule. So we have a net minus, whoops, that's a peanut in our, a net minus of 2 ATP when we are starting out this reaction. Let me get back to my little penny pen. All right, it's minus 2 ATP. Now, when we get to this molecule here, something interesting happens. Fructose 1,6-bisphosphate, we can see that there is still 1, 2, 3, 4, 5, 6 carbons here, okay? So this biomolecule, this step, we still have 6 carbons in one molecule. When we move from here to our next step, step 4 and step 5, we see that now we only have 3 carbons, 1, 2, and 3. So we go from a 6-carbon structure to a 3-carbon structure. When I see that change, I go, okay, is there any carbons that are leaving the reaction? And I don't see any CO2 being released like we do in the Krebs cycle. So what that means is we essentially break our fructose 1,6-bisphosphate molecule in half. And we create 2 3-carbon molecules or 2 3-carbon structures. And that's what's happening here. And so we didn't lose carbons. We just broke this molecule in half into 2 3-carbon structures, which will help us be able to convert into pyruvate, which is a 3-carbon structure as well. So at the end of this process, we actually ended up having 2 pyruvate molecules because we started with 6 carbons. We end with 3 carbons times 2, which equals 6. So our carbon number does not decrease. Why is this important? Because when we look through the energy generation molecules here, we can see that there's 2 ATP reactions, 2 reactions that generate ATP, phosphoglycerate kinase and pyruvate kinase. They both are the enzymes that have kinase in them that aren't included within their name. So hexokinase and phosphofructokinase, one-name enzymes require ATP versus phosphoglycerate kinase and pyruvate kinase are actually generating ATP. And because we are doing everything in this pathway twice now, because we have 2 3-carbon molecules, we actually generate 2 ATP at each of these reactions. So we have minus 2 ATP up top, and we've actually added 4 ATP in bottom, which gives us this net ATP generation of 2 overall. Same for NADH. We can see we have created 1 NADH molecule here, which actually means 2 because this whole process we're going through twice. So 2 NADH molecules as well. And that's how we get that 2 NADH. NADH molecules as well as FADH2 molecules will come into play later. These are the nicotinamide molecules, essentially. They can be converted into energy in the form of ATP when we go into the electron transport chain. So we'll kind of track these as we go through our breakdown of glucose. And then at the end, we'll see the amount of energy given. So if we open back up to our PDF, we can see starting on page 45, this is also another pathway that you can mirror. It's showing the exact same thing and the exact same reaction. It's just walking it through in a different way. But we end our process with 2 molecules of pyruvate. That's the end of glycolysis. And this all took place in the cytosol. Okay. One other important, or I guess a few other important pieces to note. Let's see here. I think those were my main ones about glycolysis. Yeah, those were my main pieces I would want you to know. Okay, so then the last component that I didn't discuss was how is our fructose 2,6-bisphosphate, as I mentioned here, a key regulatory component. How does this actually regulate glycolysis as a pathway? We have to actually see the process of gluconeogenesis to really see how that molecule regulates glycolysis, because gluconeogenesis is going to be the reverse of glycolysis. So we are going to come back to this enzyme. So I encourage you to remind me when we get here, when we get to the reverse of glycolysis, to talk about fructose 2,6-bisphosphate and how, or bisphosphate, and how it is a regulatory component of glycolysis. Before we move on to the fates of pyruvate, any questions about glycolysis? What you should know, what you can forget? Okay. All
Could
right.
you give us like an example question that it might commonly be asked?
Yeah, in the digestion of carbohydrates, what is the key rate or regulatory enzyme in the process of glycolysis? A direct enzyme question is what they would ask you most of the time for these type of questions. They also could ask you about tissue types or locations. So in the process of glycolysis or in the process of carbohydrate breakdown, what is the enzyme that governs the committal step for glucose in the liver? And that would be glucokinase. And they would probably give you glucokinase, hexokinase, phosphofructokinase, and then maybe like one other that doesn't exist. And so their key piece would be having you recognize that glucokinase is the regulatory enzyme that occurs really mainly in the liver tissue versus hexokinase is everywhere else. So if you're able to know the rate limiting steps of this reaction, so these two enzymes and kind of what they govern. So this is really the first enzyme is committing glucose to this pathway for the most part, as well as keeping glucose inside the cell, inside the cytosol, so it can't move back out, because once it converts to glucose 6-phosphate, it has a harder time leaving the cell. Versus phosphofructokinase is really your rate limiting enzyme for the whole process. So if this doesn't move forward, the process will stop at fructose 6-phosphate. So that's how they often ask these questions. They also can ask a question of what is the energy expenditure for one molecule of glucose? And then they would list something like 2 ATP, 4 ATP, 6 ATP, 0 ATP, or something like that. And your energy expenditure would be 2 ATP, but you generate 4 ATP, so you actually have a net gain of 2 ATP. So that's where those like energy questions could come into play. The last thing I see is potentially like, what's the product of glycolysis? It's more rare. Most of the time that there are more complex questions than that, but that would be pyruvate. Those are typically how I see questions. Once we get into more of our coenzymes or cofactors, we'll see how they can incorporate vitamin deficiencies and things of that nature into these biochemistry questions.
Awesome. Thank
Did
you.
that feel sufficient? Okay, great, perfect. Yeah. Any other questions on glycolysis before we move on to our next step in the pathway? Okay. So we're going to move down to our next page here. So pyruvate is our molecule that we end with at the end of glycolysis. So for one molecule of glucose, we have 2 molecules of pyruvate, and pyruvate can be converted into multiple different things. The major thing we're going to talk about first is pyruvate being converted into acetyl-CoA, because this is where, or acetyl-CoA, because this is what we're going to put into the Krebs or the TCA cycle. So this is going to continue our carbohydrate breakdown to give us energy in the form of ATP. But pyruvate does have other potential options that it can go into. Pyruvate can actually also be converted into lactate via the lactic acid cycle, alanine, or oxaloacetate. So there's essentially 4 different pathways that pyruvate can take when it is digested or broken down. And I have the pyruvate to lactate here. I don't know if I have the other pathways. I don't have those listed here right now. But just recognize that when we have pyruvate, it doesn't have to go down this acetyl-CoA pathway. It could go down using lactic acid dehydrogenase in the lactic acid cycle. It could become alanine via alanine aminotransferase, or it could become oxaloacetate via pyruvate carboxylase. So there's 4 different fates for pyruvate that it can have. Acetyl-CoA is going to be the most energy generating pathway that pyruvate can take. And so that's why we want to make sure we focus on that pathway first. There's a blank space here, and this is really for you to write down and remember the 5 coenzymes that are involved in pyruvate dehydrogenase, which is the enzyme, pyruvate dehydrogenase complex. There's 5 cofactors that are required for this complex or this enzyme. They include thiamine pyrophosphate, which is B1, lipoic acid, and I'll actually write, I can type these. I'll type them out as I talk. So thiamine pyrophosphate, which is B1. It's going to require lipoic acid. It's going to require CoA, which includes B5 pantothenic acid. It's going to require FAD, which is our B2 or riboflavin-based molecule, and NAD+, which is going to be our B3 niacin molecule. So we can see here multiple coenzymes, multiple B vitamins required for pyruvate to convert into an acetyl-CoA. So this is a treasure trove of questions when it comes to NPLEX, because they can ask you about a multitude of different vitamin deficiencies. In NPLEX II, they'll ask you even about foods potentially that you could eat or things like that to correct for these deficiencies. For the purposes of NPLEX I, they would really be giving you, they would give you this enzyme potentially, or they would say for the conversion of pyruvate to enter the TCA cycle, right? Which essentially they're saying for the conversion of pyruvate to acetyl-CoA, what vitamin cofactor is required, or which vitamin cofactor that's listed is not required. So if you're able to remember B1, B2, B3, and B5, so those are B vitamins that are needed, and then lipoic acid, that should serve you well. If you can commit to memory, B1 is thiamine pyrophosphate. That one's pretty easy because thiamine's in the name. B5 pantothenic acid is CoA, so this one maybe remember. That one would be a memorization component. FAD is flavin. And so that's the first like name, and riboflavin, right, is B2. So that's how I remember that. And then the NAD is niacin. So they both start with an N. So this is kind of a really key step. I see this tested on probably even more than glycolysis, is this pathway of pyruvate to acetyl-CoA. If you're having trouble remembering it, you could think about this mnemonic device, the lovely coenzymes for nerds. So the lovely coenzymes for nerds. As a biochemistry major in the past, that was often one of my favorites to keep these into my brain. And it will also, this complex will also serve you well when we get into the Krebs cycle, the TCA cycle, because this pyruvate dehydrogenase complex looks very similar, if not almost identical, to alpha ketoglutarate dehydrogenase complex, which is a rate limiting or a major component, major enzyme in the TCA cycle that requires the same exact 5 coenzymes. So pyruvate dehydrogenase complex, these coenzymes will come back again in the future. Okay, let me make sure I didn't miss anything here.
Dr. Petro, I have a question.
Yeah, please.
Just to orient myself, that pyruvate to acetyl-CoA takes place in between glycolysis and the TCA cycle within mitochondria?
Correct. So you can see here on this picture, that's a great question. Was that Cameron?
Yeah.
Awesome. I thought so with the voice. I couldn't see who it was. So pyruvate oxidation, we see pyruvate was created in the cytosol through glycolysis. It's transported into the mitochondria via transport protein, where then it's converted into acetyl-CoA within the mitochondrial matrix. So once we have pyruvate, it has to be transported into mitochondria to undergo pyruvate oxidation. Oh, awesome.
Okay,
Thank
thank
you.
you.
Tender loving care for Nancy. I love that too. Awesome. Thanks, Gigi. Perfect. Let's see here if there's any other ones. The energy producers of this reaction, we'll see that we get 2 acetyl-CoA molecules. So 2 of these, because we had 2 pyruvate. So we're talking energy from glucose, right? From 1 molecule glucose. So 2 pyruvate to 2 acetyl-CoA. And because we do this twice, we're going to actually generate 2 NADH. And so we get 2 more NADH added to our list. So thus far, with glycolysis and pyruvate oxidation, we now have generated 2 ATP, 2 net ATP, and 4 net NADH. So that's where we're at the end of pyruvate oxidation. And once we have this acetyl-CoA, we then can kind of move into our next step, which is going to be our TCA cycle. Before I go into there, any questions about just pyruvate conversion to acetyl-CoA and the 5 coenzymes or cofactors required?
Sorry, Dr. Petro, you said 2 NADH and 2 acetyl-CoA from pyruvate oxidation?
Correct, 2 NADH and 2 acetyl-CoA. Yep,
Okay.
so we end up with 2 of these and 2 of these because we've ran through this twice. We started with 2 pyruvate, so we're going to get 2 of everything that comes through. So 2 NADH, 2 acetyl-CoA.
Okay, thank you.
Excellent. Yep.
And is this occurring in all tissues?
This is occurring in all tissues. Correct.
Thank you.
Yep. Excellent. Good questions. Awesome. All right. So we have pyruvate. I'm going to, do I talk about, yeah. I do want to spend a moment on anaerobic glycolysis. And so I'm kind of jumping back and forth just so we can kind of see both pictures here because I think both of them are helpful. So anaerobic glycolysis occurs in the cytosol. So pyruvate oxidation occurred in the mitochondrial matrix. So we took pyruvate, we put it into the mitochondrial matrix, and then we converted it to acetyl-CoA. If we were in a state of lack of oxygenation, or if we had a cell like a red blood cell that doesn't have mitochondria, these cells can still do something with pyruvate. They can take pyruvate and they can convert it into lactate via anaerobic glycolysis. This occurs in the cytosol. So this is a different location for this pathway and this process. So if we look here, pyruvate to lactate, we are going to use the enzyme lactate dehydrogenase. That's going to be our key or our only enzyme we really care about in this conversion process. And we take 1 NADH and we're going to actually convert it to NAD. So we will use technically energy to take pyruvate to lactate. So this occurs in the tissues without oxygen in the cytosol. Your substrate's pyruvate, your product is lactate. Lactate dehydrogenase, LDH, is your enzyme, your key enzymatic step. If you have a lot of NADH present, that's going to upregulate this process because you've used some to convert it. And you're going to have a negative net energy. You're going to end up needing to use 1 NADH to convert it to our lactate. And so we can see here glycolysis, lactic acid here, and alcohol here. So these would be both of your non-oxygenation generating pathways. But lactic acid is the one that we're currently talking about when no oxygen is present. Okay, why do we even do this process? You still will get 2 ATP from glycolysis. So there still is a purpose, a method. You're undergoing glycolysis to get that 2 ATP, and then that pyruvate, instead of being converted and moving down this pathway, will be used to be converted into lactate as its byproduct. And from there, that lactate will have to be broken down in the future because the buildup can be toxic to tissues. Okay. The questions about anaerobic glycolysis typically are the location, since it occurs in the cytosol, the situation in which it would occur, places that don't need oxygen or when you are without oxygen, and what the regulation pathway is. So the fact that you're using 1 NADH. Those are typically the questions I see about anaerobic glycolysis. Okay, and here we go. Any questions about that one? That one's not as crazy. Good. Okay. All right, we're moving on down. This is just giving you an example of what these big molecules look like. Nicotinamide adenine dinucleotide, that's your NAD. Flavin adenine dinucleotide, that's your FAD. I would know the name and the acronym because you could see them write it out as the whole big thing or the acronym itself. I would not worry about the actual biochemical structures for any of these. You're not going to be given a picture of a biochemical structure. Okay. We're going to move now into our next step of our respiratory cycle. We're just moving right along. We're moving into this Krebs cycle or TCA cycle or citric acid cycle. It has many names. It again also occurs in all tissues, and we are still now within the mitochondrial matrix because remember, we took pyruvate into the mitochondrial matrix to create acetyl-CoA. And so we're going to stay within that mitochondrial matrix when we are going through our TCA or our citric acid or Krebs cycle. Our substrate that we want to start with is going to be acetyl-CoA. So we see this starting here. That's going to be our initial substrate. Our product is going to be none of the things in the circle. So I think that's really important to note is all of these kind of byproducts or steps in the circle stay here, right? This whole time. Our products that we're creating we're using this carbon molecule, we're running it through the Krebs cycle to create these products that we're going to use for energy, which is CO2, so carbon dioxide as a byproduct, NADH, FADH2, and GTP as the form of energy. And so those will be really our products that we're actually pushing out when we go through this TCA or this Krebs cycle. How many of these kind of intermediary steps do you really need to know? In a perfect world, you know all 8 of them. But realistically, I think when it comes to your exam, I would know citrate, oxaloacetate at the minimum, because acetyl-CoA is going to kind of be going into the TCA cycle, and your first step, you're going to create citrate. A lot of things happen, and there's a lot of important key regulatory steps in here. But at the end, you have oxaloacetate. That's kind of your last step before it gets converted into citrate when you add acetyl-CoA. So I would know these 3 ones at the minimum. Helpful acronym that I've found, I'll type this out if I can. Let me see. Okay, no, I don't want to annotate with this too. That's too much annotation. I'll type this out on my PDF because I can type there. Keep. So a helpful acronym to remember the key or kind of all of the actual intermediary steps for the TCA cycle is going to be citrate is Krebs, starting substrate for making oxaloacetate. So citrate and oxaloacetate obviously being the first and last. And then you can see you have citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and then oxaloacetate. So that's how you could remember all of the different intermediary steps in this process. If we're going to have key kind of rate determining enzymes, I'm going to choose 2 in this process. My first is going to be isocitrate dehydrogenase. So this enzyme here governing the conversion of isocitrate to alpha-ketoglutarate. My other one is going to be alpha-ketoglutarate dehydrogenase, which governs the conversion of alpha-ketoglutarate to succinyl-CoA. Why I've included this one is because alpha-ketoglutarate dehydrogenase has the exact same cofactors as pyruvate dehydrogenase. So it's going to have vitamins B1, B2, B3, B5, and lipoic acid. So that's such a great test question because they can ask a compare and contrast question for alpha-ketoglutarate and pyruvate dehydrogenase, or comparing the TCA cycle to pyruvate oxidation, et cetera, et cetera. Isocitrate dehydrogenase is kind of a key regulatory step because we can see here, this is the first step if we start at acetyl-CoA that's actually going to generate some energy in the form of NADH. So this isocitrate to alpha-ketoglutarate, we can see we're actually generating an NADH molecule It's the first step. So it's really our first like energy producing step in this process. We can also see that alpha ketoglutarate also produces an NADH molecule. Succinate dehydrogenase, we get our FADH molecule here. And then lastly, malate to oxaloacetate. So malate dehydrogenase, we get our NADH molecule. I don't know if you noticed, but all of the enzymes that have dehydrogenase in them are the enzymes that you're actually producing energy substances for. So when in doubt, if you have a question about what are rate limiting or energy producing enzymes in the TCA cycle, look for an answer that has dehydrogenase in it, and you'll at least be on the right pathway. Our GTP reaction is here from succinyl-CoA to succinate. That is our one molecule of GTP that we're producing. GTP like ATP. So you can kind of consider those two very similar. Okay. What are our products? What do we actually produce here, right? That's our last kind of piece of the puzzle. We end up per acetyl-CoA, we're going to get 3 NADH, 1 FADH2, and 1 GTP. So I didn't even have that on there. Let me edit that. So we have 1 GTP, but remember, we're going to run through this twice for 2 acetyl-CoA molecules per glucose. So if we look at how many times we're running through this, we had from our glucose molecule up top, we ended up getting 2 pyruvate. So we're going to have 2 acetyl-CoAs. So we're going to run through the Krebs cycle 2 times per 1 molecule of glucose. So each time we'll generate 3 NADH, 1 FADH2, and 1 GTP, which we would double, right, for the 2 times around. So 6, 2, and 2. Okay, that's our Krebs cycle.
Dr. Petro.
Yes.
So if they ask a question on the exam, like how many NADH does 1 molecule of, or does the Krebs cycle produce, would the answer be 6?
Yeah, if they asked, they would have to clarify if they're asking about acetyl-CoA, 1 turn of the Krebs cycle, or glucose, and they can ask all of those, right? So 1 turn of the Krebs cycle would be 1 time around. 1 molecule of acetyl-CoA would be 1 time around. 1 molecule of glucose would be 2 times around. So if it was 1 molecule of glucose, your answer would be, and they're asking about NADH specifically, your answer would be 6. If they're asking about 1 turn of the Krebs cycle, your answer would be 3.
Thank you.
Yeah, good question. Most of your questions you're going to get about energy generation are going to be kind of end stage ATP questions. And so we'll kind of do those conversions here in a moment, but sometimes they will ask about generation of NADH or FADH on its own. And really you just have to read the question carefully and see what they're asking for. What was their initial molecule they were talking about? What pathway or process are they discussing? And is there any clues to kind of throw you off bait, like asking about acetyl-CoA versus glucose? Okay. Where we're going with all of this is we're going to put all of our NADH and FADH molecules through the electron transport chain. This is now in the inner mitochondrial membrane of our cells. So we were originally in the mitochondrial matrix for the last 2 steps. Now we're going to move into the inner mitochondrial membrane, the innermost portion of the mitochondria. We're actually on that phospholipid bilayer. And this is where we see these 5 complexes, that we often talk about for the electron transport chain. Now, I do not suggest that you try to remember all of these 5 complexes, know their exact names, know every single cofactor. Do not do that. It's not worth your time. It's not high yield. Just know that complex 1 deals with NADH, complex 2 deals with FADH2. How I remember that is FADH2 has a 2 in it. It deals with complex 2. Okay, so that's how I separated those. Essentially, they're going to take these and they're going to generate protons. We're creating a proton gradient that's going to generate an ATPase proton pump, which will allow us to produce ATP. That's essentially what we're doing here is we're getting a lot of protons. We're creating this electrical chemical gradient using protons, using hydrogen ions that we've essentially bumped off from these molecules here. And with that proton hydrogen ion gradient, we're able to use those protons here, these hydrogen ions, pump them through this essentially complex 5, which is our pump, and take ADP and convert it to ATP plus water. And so that's really the purpose of the electron transport chain. It's to get hydrogen ions, make a concentration and charge gradient. They're going to want to go out their concentration, down their concentration charge gradient, and that's going to cause this molecule to essentially turn and produce ATP. And so that's what we're trying to do here. So like the one key molecule I'd have you know is CoQ10 is involved with complex 3. That's the one kind of more supplemental, I guess, question you could see here. CoQ10 complex 3. I would not worry about the rest of these kind of things. Okay. So this begs the question, with all of this stuff that we've just done, this is another image here. If you're more of like a visual person and like to see what the pump looks like, it kind of looks like this little cap turning thing. But at the end of the day, what is the math? And I've crossed out some numbers here because this is updated from when I initially created this with Dr. Taylor back in the day. So NADH per 1 molecule of NADH, it's going to get you 2.5 molecules of ATP. For 1 molecule of FADH, you're going to get 1.5 molecules of ATP. So we can see here in glycolysis, we had 2 NADHs, our pyruvate dehydrogenase, we got 2, and then TCA, we got 6, right? Because this is per molecule of glucose that we're talking about. We generated 2 net ATP from glycolysis and 2 net GTP from the TCA cycle. And we got 2 FADH from the TCA cycle. So these are all the things that we got that were energy creating compounds from our aerobic respiration. If we add them together, we get 10, 4, and 2, okay? Then just take 10 times by 2.5, you're gonna get 25. 4 just goes to 4. And then 2 times 1.5 gets you, oh, that's math is wrong. 2 times 1.5 is 2.5. There we go, that's better. Right, is my math right there? Yeah. Or 3, 3? 2, 1.5, 2 times 2, yeah, 3, great. Ooh, math's hard. So then we're going to end up just crossing out the wrong numbers. Then you'll see we'll end up here, 25 plus 4 plus 3. So 25 plus 7 gives us 32 ATP. And this is the most correct answer for what we now know is our energy generation from 1 molecule of glucose. 32 is pretty much the standard biochemical answer that we're looking for. Sometimes you'll only get 31 or 30 molecules, and we'll look at why here in a moment. But if you had to put one number to memory of how much does 1 molecule of glucose yield, 32 ATP would be your best, most correct answer. Any questions how I got the math? Again, I tried to write the corrections here for the conversion factor. Okay. So 32 is your best answer. Sometimes there's not 36. Sometimes there's only 30 or 31 ATP produced. So 30 to 31 or 32 is like your best answer. The reason is there's different shuttles. The shuttles are going to be what takes these byproducts through into the mitochondria. Because remember, when we created pyruvate, we did that in the cytosol, and then we had to shuttle it into the mitochondrial matrix to actually do pyruvate conversion. And so there's several different shuttles that we can see depending on the locations you're at in the body. So the malate shuttle is located in your kidney, your liver, and your heart. Your glycerol 3-phosphate shuttle is located in your brain, and your skeletal muscle. So those are kind of the general big places where you're doing a lot of aerobic respiration. Obviously, lots of cells can do aerobic respiration, but kidney, liver, heart, brain, and skeletal muscle are the areas that are doing the majority for your body. So your malate shuttle, we can see here, we're taking pyruvate. We're going to move it through inside the membrane using malate, its relationship with oxaloacetate, its relationship with aspartate. How much should you know of this? Nothing. Just know that the malate shuttle is associated with kidney, liver, heart, and you're going to move NADH with a concentration gradient. And so we can see here that to go into the malate shuttle, we're taking 2 NADH and we're converting them into 2 NAD, and then you're getting 1 NADH at the end. So you have a net loss of 1 NADH in this shuttle process. All right, because you had to take 2, actually, just kidding. You're regenerating it inside. Never mind. Net loss of nothing inside. Then we have this one, the glycerol 3-phosphate shuttle. We can see this found in the brain and the skeletal muscle. This is when we have lower NADH in the cytosol. It moves NADH against its concentration gradient. And so this is the one we're actually going to have to use some energy to move pyruvate into the actual mitochondrial matrix. And so we can see here, we use an NADH, and then we're kind of, this is circling around, this is circling around, and this is circling around. So we take an NADH, but we get an FADH2 from it, but NADH, 1 NADH equals 2.5 ATP, 1 FADH2 equals 1.5 ATP. So every time we do this, we have a net loss of 1 ATP. And given that we have 2 pyruvate molecules, we're going to have to do this twice in brain and skeletal muscle, which is how we get to 2 less ATP than normal. So in kidney, liver, heart, your answer for glucose generation or ATP generation would be 32. In your brain or skeletal muscle, your answer would be 30 for ATP. And that's kind of how we work through those problems from the back end. Okay, I know that's a lot of stuff. So what questions do we have about that little mental gymnastics we just went through? Talk to me.
For those 2 pathways, do we really just need to know like the ATP production and like where it's at, or do we need to know any enzymes or anything like that?
No enzymes. I would say not even byproducts. Just know the name of the shuttle, the location in the body, and if you're going to have to use any energy. So malate, kidney, liver, heart, no net energy loss. Glycerol 3-phosphate, brain, skeletal muscle, you're losing essentially 1 ATP per time. So 2 ATP lost total. That's what I would know. Good question.
Thank you.
Yep.
Can you repeat where it was 38 and 32 ATP again?
So no 38, yep, good
Oh.
call. So no 38, 38 would be using old numbers. So we used to say it was 3 and 2 back in the day, but now it's 2.5 and 1.5. So your answer would be 32 ATP you're generating total per 1 molecule of glucose. If you generate only 30, it's because you use that other shuttle that I previously mentioned.
Okay, thank you.
Yes, absolutely. I do believe NPLEX has updated these numbers. So they are now using these as the conversion factor. For a while, they were still using the old, which is why we had it. Okay. Awesome stuff. All right. So before we take our first break, I just wanted to have one more little moment for glucose, since that's our friend in this first half before we move on to fats, and look at what do we do when we need to generate glucose. So this is when we are going to actually be thinking about glucagon, right? Glucagon, the hormone that's released by our pancreas, is for when we have a hypoglycemic state, a low blood sugar state, and we need to get more glucose back into the blood. So glycolysis takes glucose from the blood and it converts it into energy. Gluconeogenesis takes our storage forms of glucose, whatever those are, and it's going to convert it back into glucose in the body so that we can use glucose in the blood. So gluconeogenesis, the key kind of kicker here is there's a couple different places that we can pull to essentially move into this pathway. One is using glycerol. So we can take glycerol and we can convert it into DHAP, which we can see DHAP over on this side, DHAP on this side. So really this glycerol pathway, we can use glycerol. We can also take phosphoenolpyruvate, which can come from a lot of different sources, pyruvate, lactates, alanine, amino acids, etc. So all of these components can essentially be converted up into glyceraldehyde, this other 3-carbon molecule. So really, because we have all these different ways of getting here, I want to first just focus on these last few steps, because that's really kind of the key controllers of gluconeogenesis compared to glycolysis. So for glycolysis, our rate limiting enzyme was phosphofructokinase, right? Our PFK. If we look at gluconeogenesis, one of our main rate limiting enzymes for gluconeogenesis is going to be fructose 1,6-bisphosphate, which is going to, or bisphosphatase, which is going to be literally the enzyme counterpart for phosphofructokinase. It's just taking this reaction and going the opposite direction. Okay. The other kind of secondary important rate limiting step would be glucose 6-phosphatase, which takes glucose 6-phosphate and converts it to glucose. But fructose bisphosphatase is your main rate limiting enzyme or rate limiting step here, moving from fructose 1,6-phosphate to fructose 6-phosphate. So essentially doing the reverse of this reaction. Okay, that's the biggest piece I would know here about these two pathways. Okay, let's see. The other piece that I wanted to talk about, and then we'll break, is going back up to page 43, where we talk about glycogen. Glycogen is our storage form of glucose. We'll find it in the cytosol of tissues that can store it. And it has two different bonds. There's an alpha 1,4 bond and an alpha 1,6 bond. These alpha 1,6 bonds are the bonds kind of up and down, versus the alpha 1,4 bonds are the bonds between the two glucose molecules. If we remember, do you remember which bond amylase can break versus which bond amylase can't break?
Alpha 1,4.
Sorry, alpha.
Alpha 1,4.
Alpha
Can
1,4 is the one that they can break. Yes, correct. So amylase can break these alpha 1,4 bonds between glucose molecules, but when we have essentially these chains, these alpha 1,6 bonds, amylase is unable to break those down. So just remember, like anytime you're eating a complex carb, these are the essentially the bonds that amylase cannot break down itself. The alpha 1,4s it can. So when we have glycogen, our storage form of glucose, glycogen itself can be converted into glucose 1-phosphate using glycogen phosphorylase. And then from there, phosphoglucomutase can convert either glucose 1-phosphate to glucose 6-phosphate or vice versa. This is a reversible reaction that can go both directions. And then glucose 6-phosphate can get converted into glucose using glucose 6-phosphatase only in the liver. And so this pathway, essentially from glycogen all the way down to glucose, we would call glycogenolysis, which essentially is taking glycogen and creating more glucose. So I think about it as like glycogen or glucose generation. So glycogenolysis versus the formation of glycogen, glucose to glycogen, is called glycogenesis. Okay, so I'll write those down. Glycogenesis versus glycogenolysis. So glycogenolysis, glycogen to glucose, glycogenesis, glucose to glycogen, all the way down here, gluconeogenesis, taking everything that's not glycogen and converting it to glucose. All right. Most of the time, high yield wise, when they're asking about these pathways, they try to just confuse you with those names. So if they give you anything that's not glycogen and they ask what the pathway it's going to take to create glucose, gluconeogenesis is your answer. If it gives you anything to do with glycogen and it's asking you to either create glucose or to create glycogen, then you're looking at your glycogeno or your glycogenesis instead of your glucogeno. Okay. Cool. All right. Those were the main carbohydrate metabolism pieces I wanted us to talk about. This is the glycogen synthesis process. Again, this is occurring in the liver within the cytosol. So I want to take a little bit of a break. We'll take a break for the next 6 minutes. So we'll come back together at 10. At 10 o'clock, we're going to jump gears and we're going to start moving into more of our cholesterol pathways. We're going to talk through cholesterol synthesis first, talk about that process of IDL, LDL, HDL, and then we'll go into some lipid metabolism for the next hour. Okay. All right, so I'm gonna stop sharing.
Dr. Petro, just really quick.
Yeah,
Could you repeat what you said, that last part, if they ask questions on like the glycogen versus like anything but?
yes, absolutely. So if they're asking a question about creating glucose from anything that's not glycogen, so glycerol, lactates, amino acids, pyruvate, using any other molecule as a precursor to create glucose, gluconeogenesis, hence glucose neogenesis. Yes, it will be posted after today, after we're done. But gluconeogenesis is dealing with creating glucose from non-glycogen sources. When you're dealing with the generation of glucose from glycogen, you have glycogenolysis. Glycogenolysis. Sorry, I want to make sure I spell it right. Glycogen. This is going to be how you generate glucose from glycogen, or if you're creating glycogen, so creation of glycogen, you're going to have glycogenesis. Oops. Sorry. So these three terms are they often try to trick you to get you to choose one versus the other because they are so close in verbiage. But just remember, gluconeogenesis, gluco is the beginning of glucose. So it's creating glucose from other sources. It has nothing to do with glycogen. Versus your glycogenolysis and your glycogenesis both have the beginning of glycogen at the beginning of their name. So they're dealing with the currency of glycogen. Glycogenolysis, the breakdown of glycogen to create glucose. Glycogenesis, the creation of glycogen, the storage form of glucose.
Okay, thank you.
Yeah, good question. Awesome. And I will post the recording after today. I posted yesterday's recordings already online. So once we're done today, it'll probably be recorded or posted by the afternoon. Awesome. All right, we'll break. We'll come back. Actually, we'll come back 6 after. So we'll give a true 10-minute break. Go stand up, get away from your computer, walk around, grab some coffee, whatever. And then we will come back here. I'm going to stop this recording and we'll restart our recording after the break.